Okay, I'm the next presentation entitled Characterizing Leaf Spot Resistant Loci through Image Analysis of Advanced Breeding Lines in the NCSU Peanut Breeding Program. I thought earlier this morning that Richard did an excellent job in this work, and hopefully what I'm going to present is kind of a good follow-up to what he, the brilliant work that he was doing in terms of leaf spot evaluations using drone imaging. There you go. So as we know, the Peanut Research Foundation and National Peanut Board estimate the annual losses due to early and late leaf spot to be about $53 million per year. To control leaf spot in the Virginia Carolinas region alone, it is estimated that peanut farmers spend around 80 to 90 bucks per acre, which equates to about $17 million per year when you consider average production acreage. Some of our primary concerns regarding leaf spot and resistance to this disease is the pathogen overcoming the plant resistance or maybe chemical resistance and even shifts from one pathogen to another year to year. There seems to be a limitation on novel chemistries and more of an emphasis on combination fungicides, which has the potential of allowing the pathogen to develop resistances to multiple modes of action. From a plant breeding sense, we cannot control this level of resistance, but from a host level resistance, we can identify novel resistance genes within our breeding program to help provide some protection if fungicides start to fail. In addition, if a resistance gene is only effective against one or other diseases, shifts in pathogen populations from year to year can easily overcome resistance genes specific to one or the other pathogen. Hopefully you've all heard of the Peanut Genome Initiative. The goal of the initiative in phase one was to provide a reference genome sequence of the progenitor species and cultivated species to benefit applied peanut breeders in the development, selection, and release of new cultivars. This phase was completed in 2019 with the release of the Durnensis, Ipaensis, and genomes, well, culminating in the release of the Hypogaea genome. In phase two, the Peanut Genome Initiative through the Peanut Research Foundation and National Peanut Board is they're funding 4 pillars of research, and in terms of our program and our research efforts, we're focusing on leaf spot resistance. Within our breeding program, we have varying levels of resistance depending on the resources and the specific program. In the cultivated breeding program, the one that results in cultivar releases to growers, we have a wide range of resistance and susceptible breeding lines. On the left, this is just a simple comparison of an older cultivar NCVA 92R and a newly developed breeding line N1621. So you can see where we've come so far. Relative to the performance in the cultivated breeding program, however, the wild species program holds far more resistant germplasm lines. On the right, the line in the center is a interspecific hybrid between Gregory and A. diogoi. This level of resistance is probably greater than any line in the cultivated breeding program. However, as we know with some of these introgressions, the agronomic properties can be extremely low. On an overall view of the two breeding program, there's clearly a difference in the level of resistance to leaf spot represented in these two drone images. On the left is our advanced leaf spot trial. Clearly a number of lines are brown and completely defoliated under intense leaf spot pressure with no applications of fungicide. On the right, the wild species shows consistently high levels of resistance to leaf spot under same leaf spot pressure with no applications of fungicides. And the way I know this is because these two images were taken on the same day. The main sources of resistance that the cultivated breeding program is starting to utilize or has utilized in the past stems from Arachis cardinacei or now Arachis diogoi. In the development of the cultivar Bailey, as we've seen in previous presentations in the Joe Sugg competition, a wild species introgression from the Arachis cardinacei line was used to develop one of our breeding lines that became Bailey. So we know it's possible to transfer these beneficial resistance genes into superior yielding lines in our cultivated breeding program. Many of us, our current efforts to identify and select leaf spot are somewhat limited to, kind of a subjective evaluation of breeding plots by a peanut breeder. So in this case, me. These ratings aren't as straightforward as you think and often can be compromised by changes in weather, raters, replications, field size, etc. For leaf spot, we rate on a 1 to 9 scale, where again, this is similar to the Florida scale or, somewhat modified or very similar. 1 just means resistant or we don't have any visual lesions or defoliation. 5 means we're kind of breaching that maximum lesion count to some defoliation, and then 9, which means the plot has been completely defoliated or reduced to vines. In the image to the right, I have a peanut leaf that shows symptoms of early and late leaf spot. This is obviously like a very in a perfect situation. We have early leaf spot here where we can easily differentiate versus late leaf spot. And that was kind of our goal, was to see whether or not, using some tissue analysis, if we could accurately differentiate these on a leaf level surface, and then, expand that to from a drone classification. So when we're taking ratings on a plot level, it's incredibly difficult actually to separate out early and late leaf spot percentages visually. Therefore, ratings just become an overall percentage of lesions. And then the overall importance of taking good ratings and consistent ratings is that it helps us to map these genes responsible for resistance so that we can design better genetic markers that will identify families and or lines with those important genes that we want to introduce into our program. So on the left is a representation of a single an integration block and a multiplexed Casper, in our case, PACE assay that we use for selection purposes within our program. I'm not going to cover this here, as I think Ryan has covered this in the past. But if you have any questions regarding how we're doing integration blocks, or how we're doing these multiple trait selections using a single assay, just please let me know. On the left is on the right is a simple schematic of the NCSU peanut breeding and genetics program breeding pipeline. We start by making crosses and we send them to Puerto Rico to get an extra generation of development. When the line comes back, we progeny test them in the field for the disease resistance, and then in this case, leaf spot. And we'd identify the best families from the field and pair it with our marker results, select plants with the best pods, since we're in Virginia types in those families, and then we'll repeat the cycles. And then after several cycles, these lines advance to our yield trials, and hopefully one is selected for release. So the objectives of this are based on this information. We developed a project that kind of captures these objectives. The first being is to identify objective methods of evaluating leaf spot, both early and late leaf spot, for improving phenotyping and data quality. For the interest of time and for this presentation, we're only going to focus on this first objective, although the other ones are very important in mapping and identifying either introgression regions or any kind of regions within our advanced pipeline for selection purposes. The data that I'm going to show today represents some of the preliminary work that was conducted in 2019 and 20. In 2019, we grew out some representative samples of commonly grown cultivars within the breeding program leaf spot trial, and they were assessed using developed scripts in Fiji. So this is well before field image R came out, so these scripts had already been developed. And we were trying to use those to separate visual ratings into their subcomponents. So what we wanted to know was, could you effectively scan the tissue collected and differentiate between early and late leaf spot using the images alone? And in addition, could we use those tissue scans to estimate an overall percentage of leaf spot infections on leaf tissue? And then number two, could we track using UAV imaging over time the relative canopy losses due to leaf spot infection that could be attributable to defoliation? And as we know, Richard showed this previously, so we know that this can be accomplished. So to do this work, we selected 8 leaves randomly per plot, and then the 32 leaflets were scanned on an Epson workforce scanner for analysis, or for tissue analysis. And then the canopy cover was captured using a DJI Mavic 2 Pro drone, which comes stock with a 1-inch sensor, and images were stitched together using Agistoph Metashape. So it turns out it's way more difficult to separate these diseases out using basic thresholding. The issues include whether each leaf spot can be accurately identified and how long it takes to go through each leaf sample to identify the specific disease causing each spot. We can also have some issues with resolution in the image, and it may not allow us to accurately differentiate the spots. Third could be thresholding cutoffs that sort the clusters or leaf spots may be too similar. And then population pressure of each disease in a given year may change. So for example, in 2019, it was estimated that we were around 95% late leaf spot. So clearly it's very difficult to train any kind of model on picking up early leaf spot when the population pressure is so low. So in order to attempt at differentiating these diseases, more data needs to be collected on the specific leaf spots in order to develop better training models for accurately classifying early and late leaf spot infection. That being said, getting a percentage of leaf spot area is pretty straightforward. The Fiji script is designed to threshold the leaf spot and the entirety of the leaf surface separately, and then the ratio between the 2 pixels gives us our total leaf spot estimate on the leaf surface. Similarly, the drone images help us to estimate canopy cover changes during heavy leaf spot pressure. The top and bottom rows in the images were taken on different days, so September 6th versus October 8th prior to harvest. And then these columns just kind of show the difference in thresholding. And so we can grid these plots out. I just did a quick one on this single plot, and then how we kind of gather our estimate is just a difference in the 2 canopy estimates. And so using these 2 scores, we can get the change in the canopy over time, which is what we're calling defoliation. And in some instances, when a plot is relatively unaffected by leaf spot, the defoliation score can be negative due to continual or unaffected growth. We can then apply this method to our entire breeding program. In the field, we grew out 220 lines in 2020 and used this approach to get leaf tissue and defoliation estimates for each plot on a biweekly basis during heavy leaf spot pressure. We additionally have yield data collected on these untreated plots, along with some plots that were treated with fungicide, but we're not going to present that here. So just some results on what we got here. So presented are what we obtained from the previously described methods in 2020. On the left is a figure of 26 lines that were whole genome sequenced for another project in which we're developing a high throughput genotyping platform for our program moving forward. These lines are chosen based on several traits of interest, including resistance to leaf spot. These differences in the bar chart represent the visual ratings taken on the genotypes prior to harvest. On the right are some basic statistics describing the different evaluation methods, including the tissue scans alone, the drone or defoliation score alone, or a combination of the 2 using the first principal component when compared to visual scores. In each instance, the evaluation type was highly correlated with visual ratings and concurred with ranking as well. In addition, the visual score 1 to 9 was also modeled using a logistic regression to determine the predictive potential of each evaluation type. The logistic model produces overall concordance scores, which is the model's predictive potential and odds ratios for each evaluation parameter. The concordance scores compares pairwise scores across the data set to determine if the probabilities associated with being in a higher visual score are true for each pair that's tested. For the odds ratio, this is determining the odds of being in a higher visual score given an increase in the evaluation unit. So if you look at the models evaluated, tissue alone, drone alone, PC1, and the full model of tissue plus drone, there's an increase in concordance when considering more parameters, but it seems the PC1 that explains about 79% of the variation is similar to the full model. That being said, there isn't much of a difference in concordance when using tissue or defoliation data alone. And I think this is kind of a summary of what Richard was showing earlier. For the tissue, there is 1.3 times the odds of being in a higher visual score when the percentage of leaf spot surface area increases, which makes sense. And then under the PC, we're 10 times more likely to be in a higher visual score when we look at the two components together as they're increasing. Our future work on this leaf spot evaluation method is funded through the USDA NEFA program, and we're working in the next field days, or field season will be conducted by my PhD student, Cassie NEWMAN. The work we're doing in the field this year consists of multiple ratings, timings, data collection types, and we'll be evaluating the plots for leaf spot on a previous 1 to 9 scale. This will be done in a weekly manner. Cassie will be evaluating the plots using what we're calling our low throughput objective method of drone imaging for defoliation and tissue collection for lesion counts and leaf spot differentiation. And then lastly, we'll be collecting high throughput images and developing an algorithm to rapidly analyze the images for instant leaf spot ratings using cell phone capture. The collection time will be about August through October. Here we're diagramming our high throughput method that uses drone or cell phone images to rapidly take ratings on both defoliation and leaf spot lesions. The idea here is to evaluate these 200 breeding lines or 200 plus breeding lines in the field using the drone imaging and walking through the plots taking cell phone images. The images will be uploaded to where image software with a customized algorithm will be waiting to analyze the plots. In addition, we're going to evaluate using Canopio to determine the effectiveness of this application in detecting leaf spot lesion counts and canopy changes. So hopefully using these ratings, these can be taken objectively in less time than the breeder walking the entire field. And if the analysis is complete, I can go ahead and then look at best lines and making selections within these lines for further development. And then ultimately, we're hoping to achieve is to pair our newly developed genotyping pipeline to high quality leaf spot ratings in order to identify genes that are influencing leaf spot resistance. And so with that, just like to acknowledge some of our funding sponsors. And at this time, well, I guess since I'm the moderator here, I guess we're going to move on since we don't really have that much time in terms of questions.